Welcome to this rigorous scientific lecture on one of the most fascinating examples of plant defense chemistry. Today we're going to explore the molecular mechanisms behind why onions make you cry. Why does cutting an onion cause lacrimation or tearing, while simply holding an intact onion does not? This question leads us into a beautiful cascade of enzymatic reactions, physical chemistry, and biological defense mechanisms. In an intact onion cell, there are two key components that are physically separated. First, we have sulfur-containing amino acid derivatives stored in the cytoplasm. Second, we have an enzyme called alliinase stored in the vacuole. Here's a simplified diagram of an intact onion cell. The cytoplasm contains sulfur-containing compounds, while the enzyme alliinase is safely stored in the vacuole. As long as these remain separated, no reaction occurs. When you cut an onion with a knife, you physically rupture thousands of cell membranes. This disruption is critical because it brings our two separated components into contact for the first time. The knife blade ruptures the cell membranes, and suddenly the cytoplasmic contents mix with the vacuolar contents. Now the enzyme alliinase can access its substrates. Once the enzyme and substrate meet, a remarkable cascade of reactions begins. Let me walk you through this step by step with chemical precision. The enzyme alliinase acts on sulfur-containing amino acid derivatives, specifically SALKNY-LL-cysteine sulfoxides. This is the first step in the cascade. Alliinase plus sulfoxide produces sulfenic acid as the primary product. But the reaction doesn't stop there. Here's where it gets interesting. A second enzyme, called lachrymatory factor synthase, or LFS, immediately converts the sulfenic acid into the actual tear-inducing compound. The sulfenic acid is converted by LFS into syn-propanethial S-oxide. This molecule is the actual lacrimatory factor, the chemical that makes you cry. Let's examine the properties of this remarkable molecule. Syn-propanethial S-oxide has several key characteristics that make it so effective at inducing tears. First, it's highly volatile. The molecular weight is approximately 90 grams per mole, which makes it light enough to readily evaporate at room temperature. This volatility is crucial for the molecule to reach your eyes. Second, it's electrophilic. The sulfur-oxygen double bond creates a partial positive charge on the sulfur atom, making it reactive with nucleophiles. Third, it's somewhat water-soluble, which allows it to dissolve in the tear film on your eye surface. This is essential for its irritant properties. Here's a simplified representation showing the key sulfur-oxygen bond. This molecule is small, volatile, and reactive, the perfect irritant. Now let's discuss how this molecule travels from the cutting board to your eye. This involves principles of diffusion, convection, and fluid dynamics. Let's ask ourselves a question. How quickly can this molecule travel from the onion surface to your eye? Let's do a back-of-the-envelope calculation. We can estimate this using diffusion principles. The diffusion coefficient for small organic molecules in air is approximately 0.1 square centimeters per second. The diffusion coefficient d is approximately 0.1 square centimeters per second. The characteristic diffusion timescales as L squared divided by d, where L is the distance. If the distance from cutting board to eye is 30 centimeters, pure diffusion would take 9,000 seconds or about 2.5 hours. But we know it happens much faster. The answer is that convection dominates. Air currents, your breathing, and thermal convection from your body create bulk flow that transports the molecules much faster than diffusion alone. Typical air velocities in a room are 10 to 50 centimeters per second. At 20 centimeters per second, the molecule reaches your eye in just 1.5 seconds. Time equals distance divided by velocity. 30 centimeters divided by 20 centimeters per second equals 1.5 seconds. This matches our experience. When syn-propanethial S-oxide reaches your eye, it dissolves in the tear film and triggers a sensory response. Let's examine this mechanism. The tear film covering your cornea is remarkably thin, only 3 to 10 micrometers. This thin layer allows rapid diffusion of the irritant to nerve endings. Sensory neurons in the cornea express TRPA1 receptors. These are transient receptor potential anchorin 1 channels that respond to electrophilic compounds, like our lacrimatory factor. The irritant molecules dissolve in the thin tear film and activate sensory neurons. This triggers the lacrimation reflex. The signal travels via the trigeminal nerve to the brainstem, which then sends signals to the lacrimal glands to produce tears. This is a protective reflex to flush out the irritant. Now let's address a practical question. Why does chilling an onion reduce tears? The answer lies in fundamental thermodynamics and kinetics. Let's ask, what happens to the enzyme activity when we chill the onion? There are two key effects. First, enzyme reaction rates depend on temperature, according to the Arrhenius equation. Lower temperature means lower kinetic energy and slower reactions. The rate constant K equals A times e to the negative EA over RT, where EA is the activation energy, R is the gas constant, and T is temperature in Kelvin. For most biological reactions, the rate approximately doubles for every 10 degrees Celsius increase. This is called the Q10 rule. Q10 approximately equals 2. If we chill an onion from 20 degrees Celsius to 4 degrees Celsius, that's a 16-degree drop. This reduces the reaction rate by approximately a factor of 3. The ratio of rate constants is approximately 2 to the power of 1.6, which equals about 3. The second effect is even more important. The vapor pressure of the lacrimatory factor decreases exponentially with temperature. Vapor pressure P equals P0 times e to the negative delta HVAP over RT, where delta HVAP is the enthalpy of vaporization. For typical organic molecules, vapor pressure roughly halves for every 10 degrees Celsius decrease in temperature. So chilling from 20 to 4 degrees Celsius reduces vapor pressure by about a factor of 3. Combining both effects, slower production and lower volatility, we get approximately a 9-fold reduction in the rate of irritant delivery to your eyes. This explains why chilled onions cause fewer tears. Now let's discuss why cutting technique matters. The key is minimizing the number of cells you rupture and maximizing the speed of cutting. A sharp knife makes a clean cut through cells, rupturing only those directly in the blade path. A dull knife crushes and tears cells, damaging many more cells in the vicinity. With a sharp knife, you get a clean vertical cut with minimal cell damage. A dull knife causes extensive crushing and tearing, rupturing far more cells and releasing more lacrimatory factor. Let's estimate. How many cells does a single knife cut rupture? We need to know the cell size and blade thickness. Typical onion cells are about 0.1 millimeters, or 100 micrometers in diameter. A typical kitchen knife blade is about 0.5 millimeters thick near the cutting edge. The number of cell layers ruptured is approximately blade thickness divided by cell size. 0.5 millimeters divided by 0.1 millimeters equals 5 layers. For a cut that's 5 centimeters long and goes through a 1 centimeter thick section, you rupture about 5 times 50 times 100, which equals 25,000 cells. A dull knife might rupture 10 times as many. The speed at which you cut also affects the concentration of irritant near the cutting board. Fast cutting creates more turbulent airflow. When you cut slowly, the lacrimatory factor accumulates in the boundary layer near the cutting surface. This is a region of relatively still air where diffusion dominates. Fast cutting with the knife creates turbulent eddies and mixing. This actually helps disperse the molecules away from the concentrated region near your hands and face. In the slow cutting scenario, molecules rise slowly in the boundary layer and drift toward your face. The concentration remains high near the surface. The optimal technique combines a sharp knife for minimal cell damage, fast cutting to create turbulent mixing, and good ventilation to remove molecules from the area. Some people cut onions under running water or in a bowl of water. Let's analyze why this works from a transport perspective. Syn-propanethial S-oxide is partially water-soluble. When produced, it can dissolve into the water rather than evaporating into the air. Let's ask, what fraction goes into water versus air? This is described by the partition coefficient. The partition coefficient K equals the concentration in water divided by the concentration in air at equilibrium. For moderately polar organic molecules like our lacrimatory factor, K is typically between 100 and 1000. This means water is far more effective at capturing the molecules. Additionally, a layer of water on the onion surface creates a physical barrier. Molecules must diffuse through the water before reaching air, which dramatically slows transport. Diffusion in water is about 10,000 times slower than in air. D water is approximately D air divided by 10,000. Now let's step back and ask the evolutionary question. Why did onions evolve this defense mechanism? What is the adaptive advantage? The lacrimatory factor serves as a chemical defense. Herbivores that bite into an onion experience immediate irritation, which discourages further consumption. The sulfur-containing compounds also have antimicrobial properties. When cells are damaged by fungal or bacterial attack, these compounds are released and inhibit pathogen growth. The clever aspect is that these toxic compounds are stored as safe precursors. Only when cells are damaged does the enzyme cascade activate, producing the defensive chemicals on demand. Let's try to quantify the tear response. How much lacrimatory factor is needed to trigger tearing? Studies suggest the threshold concentration in air that triggers lacrimation is approximately 1 part per million by volume, or about 3.7 milligrams per cubic meter. C threshold is approximately 1 part per million, which equals about 3.7 milligrams per cubic meter. How much total lacrimatory factor can one onion produce? Let's estimate this. A medium onion has a mass of about 150 grams. The sulfur-oxide precursor content is roughly 0.1% by mass. That's 150 milligrams per onion. Mass of precursor equals 150 grams times 0.001, which equals 150 milligrams. Not all precursor converts to lacrimatory factor. Assuming 50% efficiency, we get about 75 milligrams of active irritant per onion. Mass of lacrimatory factor is approximately 75 milligrams per onion. If uniformly distributed, this 75 milligrams could reach threshold concentration in about 20 cubic meters of air. That's roughly a 3 by 3 by 2 meter room. Volume equals 75 milligrams divided by 3.7 milligrams per cubic meter, which equals approximately 20 cubic meters. In practice, ventilation and diffusion mean most of the lacrimatory factor escapes before reaching this concentration. But this calculation shows one onion has plenty of irritant to affect anyone nearby. Modern biotechnology has produced a solution, tearless onions through genetic modification. Let's understand how this works. Researchers have created onion varieties where the gene encoding lacrimatory factor synthase is silenced using RNA interference. The alliinase still acts on the sulfoxide precursors, producing sulfenic acids. But without LFS, these sulfenic acids spontaneously rearrange into non-volatile thiosulfonates instead of the lacrimatory factor. Sulfenic acid converts to thiosulfonates, which are non-volatile compounds, no tears. Interestingly, thiosulfonates contribute to onion flavor, so these tearless onions retain much of their characteristic taste without the irritation. Let's summarize our findings with practical recommendations based on the science we've discussed. Chilling to refrigerator temperature for 30 minutes reduces both reaction rate and vapor pressure by about 3-fold each, giving a 9-fold reduction overall. A sharp knife minimizes cell damage and reduces the total amount of lacrimatory factor released. Ventilation creates convective flow that removes molecules from the cutting area before they reach high concentrations near your face. Water dissolves a lacrimatory factor and creates a diffusion barrier, preventing vapor transport to air. Physical barrier protection is nearly 100% effective. Goggles prevent any molecules from reaching the tear film. Genetically modified tearless onions eliminate the problem at the source by preventing lacrimatory factor synthesis. Combining methods yields multiplicative benefits. Chilling plus sharp knife plus ventilation can reduce tearing by more than 20-fold. The onion's tear-inducing mechanism is an elegant example of chemical ecology and defense biology. We've seen how cellular compartmentalization keeps reactive components separated until needed, how a cascade of two enzymes produces a volatile irritant on demand, how physical chemistry governs transport from cutting board to eye, and how temperature affects both kinetics and thermodynamics. This system beautifully demonstrates principles from organic chemistry, enzyme kinetics, transport phenomena, thermodynamics, and sensory neurobiology. It's a wonderful case study in how evolution produces sophisticated chemical solutions. Thank you for your attention to this rigorous exploration of why onions make you cry. The next time you shed a tear while cooking, you'll understand the molecular mechanisms at work.